welcome everybody. Welcome to this special sort of virtual version of the MGMS tour. For those of you who don't know who I am, my name is Phil Biggin. I'm the current chair of the Molecular Graphics and Modeling Society. And we are the ones who are kind of running this particular seminar today. So welcome to everybody. And it's my great pleasure to welcome Rommie Amaro to give this talk today. And I'll just give you a little bit of brief introduction to Rommie, just so you know a little bit about her before she speaks. So Rommie obtained her first degree, I believe, in chemical engineering from the University of Illinois at Urbana-Champaign in 1999, and then spent a couple of years, I think, as a research engineer at Kraft Foods, and then decided to come back and do a PhD with Zaida Luthi-Schulten, again at UIUC, University of Illinois at Urbana-Champaign, attending a PhD in chemistry in 2005. And then she did PDRA with Andy McCammon at UC San Diego. And that was funded by an NIH Kirchstein National Research Service Award. And then she joined the faculty of University of California at Irvine in 2009, with joint appointments between pharmaceutical scientists, computer science and chemistry, and received a couple of key awards there, I believe, the NIH Director's New Innovator Award in 2010 and a Presidential Early Career Award for Scientists and Engineers. She then returned to UCSD in 2012, where she's been ever since, and was the director of the UCSD's National Biomedical Computational Resource from 2014 to 2020, and was co-director of the Drug Design Data Resource, or D3R, as well. More recently, you may have come across some of the work, and I think she might say something about this work. That was awarded the prestigious Gordon Bell Special Prize for High Performance Computing-based COVID-19 research. So hopefully we'll hear something about that later on in the talk. So without further ado, I'll hand you over to Rommie. But before I do that, just to remind everybody just to make sure you're muted throughout the talk. And then there'll be a chance at the end to ask some questions. But if you want me to ask them on your behalf, or Ava to ask them on your behalf, then please just post them in the chat, and we're quite happy to read them out if you prefer not to reveal yourself, as it were. So without further ado, I'll hand over to you, Rommie.
Thanks, Phil. Thanks for that nice introduction. I actually probably won't be talking too much about the Gordon Bell work. Yeah, I guess I could figure out a way to mention it, but I had planned to talk about some newer work, actually, on what I've entitled "In Situ Dynamics Reveal Unseen Vulnerabilities of Viral Glycoproteins." So I'll go ahead and get started. As he mentioned, I'm going to try to keep this to about 45 minutes, so we have some time for discussion at the end. Okay, let me do that again. Okay, so hi everyone. I think I probably know many of you, but for those who I don't, so again, my name is Rommie. I'm from UC California, San Diego. And the work in my group is entirely computational and theoretical, and we really are very excited to use computational techniques, particularly multi-scale computational techniques, as essentially as a computational microscope. And the main method that I'll discuss today, and really the main one that we use and develop in the group, is one that I'm sure many, if not all of you, are already familiar with. These are these all-atom molecular dynamics simulations, which I think a wonderful analogy of those are to use them as sort of a computational microscope. And the idea is that nowadays we have tons of really fantastic biomedical data sets, and this could be different types of structural data sets taken at different resolutions. So things like high resolution X-ray crystallography, single particle cryo-electron microscopy has been fantastic, of course, and also techniques like tomography to give different views of biological structure at different scales. And we can now combine those data sets together with information from, again, really horrific techniques, things like glycomics, lipidomics, and genomics, in order to create these highly detailed three-dimensional models of biological systems. And once we have those models constructed, and I'll talk a bit about the construction of those models in the talk today, but once we have those models constructed, essentially all that we're doing is approximating that sort of complicated system down to its many atoms. We have our potential function, of course, which I'm showing here, which basically describes the interactions of each atom with all the other atoms in the system. And then all we're doing, as many of you know, is basically integrating Newton's equation of motion over time. And nowadays, thanks to advances in graphical processing units, as well as the continued growth of the supercomputing platforms, we have these really terrific engines on which these dynamical simulations are generated. The work that I'll talk about today has primarily been supported by the national supercomputers here, one in Texas called Frontera, which is just a fantastic machine. And then another called Summit, which is a Department of Energy machine at Oak Ridge National Lab. But in any case, what I hope to convey through the course of the talk is how we're using really these data-centric computational simulations to not only integrate these different types of data sets, but also then, I think more excitingly, of course, is really to extend where we can go with experimental data and to really give atomic scale views into cellular scenes. Okay, and so by way of background, so about 10 years ago now, or just about 10 years ago, we're very interested in trying to use molecular simulation as a tool that would enable computer-aided drug design. And first we really focused on small molecule discovery. And as I go through the talk, what I hope to convey also is how we're now using these same types of approaches, but to give insight into structure-based vaccine design and understanding of immunogens. But just to take a little brief sort of review of the past. Some number of years ago now, we became very interested in one of the first targets we became very interested in as a proof of concept for using simulations in drug design was this molecule P53, which I'm showing here. And what we were doing was basically trying to use molecular dynamic simulations to characterize the target flexibility, right? So we have these wonderful high resolution structures from X-ray crystallography, but they give us these static snapshots. And even when, in the case of P53, you have so many different groups who are interested to study it, you have even hundreds of different crystal structures in the protein data bank, for example, that many of those, often they're each of them anyway are only capturing sort of like one sort of state or snapshot, energy minimum of the particular molecule. And they give sort of an insight into the ensemble, but for the case of P53, most of these structures sort of look the same. So we started simulating it, and this is sort of the wiggling and jiggling of the atoms. This is sort of a typical output from these molecular dynamic simulations, again, which I know you're all familiar with. We became really interested. One thing we noticed for this particular molecule, so I'm showing here in purple the DNA binding domain of P53 in purple ribbon, and then with that green surface representation, what I'm showing is basically a little bit more of the surface, and there's one cysteine there in the center that I've sort of highlighted. And we became interested in this particular cysteine because we saw that even after some short timescale dynamics, even just on the order of hundreds of nanoseconds, we were able to see a relaxation and structural flexibility of this molecule that was not indicated in the crystal structures. And so, one of the reasons why people care about this is because trying to reactivate P53 has been a longstanding dream for cancer therapy. So P53 is known as the guardian of the genome, and it's implicated, mutations of P53 are implicated in over 50% of human cancers. And so it's a very important transcription factor that ties to a number of different downstream pathways related to cell apoptosis and so forth. And so what happens is that you have P53, when it's mutated, basically is inactivated, and it's inactivated because it's essentially destabilized, and then it can't bind the DNA. And so what we had just tried to do was basically understand how we could reactivate P53 by binding of small molecules that would restabilize this domain and allow p53 to bind to the DNA and and do its normal business to activate those pathways that would kill the cells. And so, so what we found early on in the dynamics was that, if you looked at all of the available crystal structures, and again, there were some hundreds of them in the PDB data bank, what you saw in this particular region that I was highlighting previously, it looked the same across almost all the mutants that were available, as well as their quote unquote wild type structures, which were still heavily mutated, by the way. But there was this small area that was pretty inaccessible to solvent. But what we saw after, as I was showing, just some tens of nanoseconds, hundreds of nanoseconds of dynamics, was the relaxation and opening of a pocket in this area. And so we saw this new site opening, and this new site, once you obtain these sites, there's any number of ways to interrogate how druggable or how ligandable they are. We used at first computational solvent mapping, which is sort of the the computational analogy to solvent crystallography, or cocktail crystallography, where basically you have this, you can take different structures of your molecule, flood the surface with different organic probes, and then evaluate the interaction energy of each of the probes, with the surface of the protein. And that identifies, so we did that using a wonderful tool that was created by Sandor Vaja at Boston University, called FTMap. We still use that. It's a great tool. And it's just remarkably predictive in terms of finding sites on proteins that are capable or favorable for binding ligands. And so when we investigated that site, what we saw that it was actually a hotspot for binding. This new pocket that opened was in fact a hotspot for potential ligands. We then did drug docking or small molecule docking, virtual screening into that pocket, and this sort of laid a path in terms of the methods for this concept of trying to identify these pockets that are unseen in experimental data, and then trying to translate that new structural understanding to develop small molecules that might be useful either as chemical probes, or in the ideal case as eventually potentially in patients. So with this new structural understanding, and after some virtual screening using Autodoc Bina, this was now some years ago, we basically passed, some small, actually relatively small number of compounds to our experimental colleagues for testing to see if any of them would reactivate p53. What we found was one molecule here called stictic acid, which we weren't necessarily identified in any kind of lead compound. This is a natural compound that we found bound to p53, the DNA binding domain, as well as showed dose-dependent rescue in mammalian cancer cells. So this molecule would bind to the DNA binding domain and restore the stability of these cancer mutants in p53. And the other interesting thing, I think, again, more interesting even than this particular molecule, was that this understanding of the structure allowed us to explain the mechanism, or provide the mechanism of action for a number of other reactivation molecules that had been put forth into the literature by folks like Elm Fairst and other colleagues. And so there were a number of cysteines, and due to technical reasons, it was difficult for them to find out in vivo which of the cysteines were the ones that were reactivating these covalent molecules that others had found. And through this discovery of the flexibility of this pocket, we narrowed it down to one particular cysteine that I was showing you, which is this one at position 124. And we were able to show that these reactivation compounds, such as Prima-1 and other derivatives, were actually acting through reactivation at that particular site. So with that knowledge in hand, we then significantly expanded. We started with a small library, and then one can imagine sort of waging a whole campaign. We actually formed a company called Actavalon, which has recently gone defunct. But there's still some interesting science that we were able to do there in trying to, move these molecules from sort of proof of concept, into something that might be able to get into to people. But what we were able to do is also sort of generate a computational workflow that was able to discover more novel reactivation compounds in a pretty rapid time frame, in a number of months, versus like all the research efforts of the 20 years combined. And that was really because we had this sort of novel structural understanding that we then could really sort of use as the guiding principle for this work. And so we were able to find a whole number of different compounds. Actually, there's a lot of chemical matter here. I think most of this has been disclosed, but all of these molecules were able to rescue p53 activity and kill cancer cells. More interestingly, just a few months ago, so now with Actavalon shuttering its doors, there's some interesting IP that we're basically sort of, getting out the door now. And so one of them, the first paper was this coming out was this, I think, a sort of comprehensive paper in cell chemical biology, where we showed that this particular pocket that we found, it not only exists in the wild type p53, but it also exists in several of the different mutants. So p53 has a number of different mutants that are sort of commonly found in human cancer cells, and it's druggable. This particular pocket, one of the things that was sort of intriguing about it was that it existed across these mutants and was druggable in them also. So, it had this high therapeutic potential, which is how we we're able to convince folks to invest in the vision of the company. And after some years, what we were able to show was that these compounds actually did, so we were never able to get, and we're still unable to get, an actual high resolution structure of any of these molecules bound to the target, believe it or not. But that we did every other type of biophysical assay that one could imagine to try to really tease apart or delineate whether or not these molecules were actually, acting in the way that the simulations had predicted. And pretty much every single assay that we performed indicated that these compounds in our series restored chromatin binding. So they restored this, and they also, in doing so, it was implicated that they restored the structure back to the wild type, but we also could investigate that through binding of antibodies that specifically recognize the wild type structure. And we were able to also replicate that with these antibodies. And then I'll just say, we looked at treatment in these sort of mouse models of tumors, and we saw that when we reactivated them, these tumors shrank and so forth. So that work has recently gone out. But this is really, this isn't a talk about p53. You're probably wondering, when is she going to talk about viruses? I'm getting to that. It's really just to sort of lay the storyline that, initially we sort of start with these sort of small single structures to try to use simulation in a way that, could drive experiment, particularly in a space of trying to discover new chemical matter. And of course, this, as we all know, I think as many of us know, this is now something that many folks are trying to do across a variety of targets for various indications, from viruses, as I'll talk about, to cancer and other things. And one just really beautiful, I think, nice example from just a few months ago in June, David Shaw, one of our wonderful colleagues in New York, has really dedicated a huge amount of his effort and funding portfolio to also use molecular dynamic simulations in this way. And they recently had this very nice deal with Eli Lilly for these first-in-class therapeutics against this ion channel, where they haven't released too much of the actual data around in the science story, but there was a rather large, financial deal targeting, with the exchange of IP, where Eli Lilly is going to go after trying to further commercialize this. So that's actually a really, I think, wonderful advance for the field, showing that we can really use this as a sort of a method, these simulations as a method to generate small molecules or chemical matter, and really to do it in a way that you can have potentially real impacts. That's terrific. And this is just one example. There's certainly a number of them. Okay, but I'm an academic and like to do things. There is definitely, I think working in industry and driving towards that goal is like a fantastic thing. We also like to try to do new things. That's wonderful, the freedom that we have as academics, right? So what we've long wanted to do is sort of try to move beyond just this idea of simulating a single protein, like we did, or domain even of a protein, like we did with p53, but really try to understand what that. So this is a picture of a ribosome shown as a blob. Really try to understand, what's actually happening in cells. And to do that, one has to, I think, really get a better grip on all of the complexity, right? I mean, we are reductionist folks in nature. You know, our domains or our disciplines tend to be rather reductionist, but this is certainly something that, you know, keeps me up at night, I would say, is trying to really understand what's going on so we can better understand off-target effects, so we can better understand, you know, what where these molecules are and what they're doing when and so forth. So, you know, on the way to that, you know, it quickly becomes apparent that if one wants to understand what's happening at cellular and higher levels, you know, you can't, I mean, one could, I guess, imagine, you know, building some all atom model of a cell and simulating it, you know, I don't know how long you're ever going to get. Maybe if they come with quantum computing and so forth, or neural computing, we'll be able to go out for long time scales. But, you know, you really need a multi-scale approach, probably even then. And also, you know, you probably don't want to know where every hydrogen is moving or where every, you know, atom is across life scales, right? You know, or the time span of an organism's life. So we have these different ways, you know, and I think we should really focus on developing methods, of course, that get us out to longer time and length scales where we can sort of, you know, switch the physics as needed in order to do that. So, you know, we've been really interested to develop and link to larger scale time and length scales through multi-scaling. And I think computational biophysics in general is very well suited to doing that. And there's these really beautiful examples that we see from many groups. One area, so there, I guess I'll say that, you know, we're really good in general at simulating particular scales or having methods that interrogate particular scales, such as MD to look at the molecular and macromolecular scales, subcellular studies, things like reaction diffusion models and so forth. At the cell scale, typically we're talking about biomechanics type methods and so on, right? You know, there are tools that are really capable within each domain, but where it gets harder is really crossing the domains and combining them to get this sort of hierarchical understanding of the inner workings of cells, all the way down to, I mean, and one of the things that I guess we're really excited about, because you see, I never really let go of all those atoms, but we're really excited about sort of, you know, linking to those higher level models, but always with the ability to come back down to the motions of atoms at the molecular level, you know, or the molecules essentially, so that we can ultimately, you know, ideally actually design chemical matter that could affect behavior across the scales. So I guess where we've really been pushing in my group is sort of at this, what I would call gap that exists between the molecular and macromolecular and subcellular scales. You know, much of our work in COVID that I won't talk about, you know, with aerosols, for example, gets us to these sort of micron level scales or near micron scales, and longer, you know, time scales, also using enhanced sampling techniques. But anyway, this is the key zone, which I'll talk about now. Okay, and so just a shameless plug for a paper that, you know, I'm still sort of proud of, which is this one that Adrian Mulholland and I wrote together some years ago now, in, you know, and how these multi-scale methods can be used, particularly with an emphasis on in drug design, to really bridge chemical and biological complexity. And, you know, it's really, I think still, I still wake up every day and think, gosh, it's exciting to be a computational biochemist, biophysicist, chemist, however you label yourself, because we have, you know, such terrific folks working to improve the algorithms and the methods, as well as folks working to, you know, make better compute architectures and the data sets that we're getting from experiment, which, you know, are integrated in such a key part of the models that we're developing. Okay, so so we have worked for a number of years to develop the tools that allow us to go from the single protein to these larger scales, such as one of our sort of leading examples has been with viruses, and actually, but not with SARS-2, although we've done, you know, a fair number of work there now, sort of in a surprise turn of events. But also with the flu or influenza. And so, you know, the flu data had been very good flu data had been around long before, of course, we had all the data now that we do for SARS and SARS-2. But we basically developed a number of tools such as Pine Molecule and Lipid Wrapper, and also CellPack, which was developed at the center that I directed, not by me. This is by folks at Scripps, such as Art Olson and David Goodsell. But basically tools that allow us to integrate high resolution, for example, X-ray or single particle cryo-EM structures of receptors into these tomographic maps. These are tomograms, these are relatively lower resolution maps of the flu that you can see here, right? And so here I'm showing the construction. But what's, what I hope to convey is that basically these simulations that are, large scale in terms of like the number of components, that they really open up new avenues of investigation that are not possible on the single protein scale. And so one thing I'll mention, so our first work in the flu, it did not actually contain the N and O linked glycans, which we later learned, we really learned are so important for the function of these glycoproteins. And so one way that we learned that, this is like I think this is pretty much the only thing I'm going to say about the Gordon Bell work, but is that, all of us were affected, of course, by COVID-19, which is the disease caused by the SARS-CoV-2 virus. And, on the outside of the virus is a spike protein, which is really important because it's like the first point of contact that human cells have with the virus. And so it plays a key role in the infection process. So they had relatively high resolution cryo-EM structures that basically gave the positions of all of the atoms in the protein, as well as the first N-link glycan sugar unit. But we were using the beautiful glycomics data coming from, for example, the group of Max Crispin, also Faris Duazadi. There's a number of groups throughout the world who are producing these data. These data sets basically give us the molecular recipes of each of these N and O link glycan sites. And so in the case of SARS-2, what we learned was that, this spike protein was really covered in these glycans, and that it was really important that we included them in the simulations. And so on the left, what I'm showing is the protein without glycans. This is essentially what the structural biologists mostly see, because the glycans are moving around too much. And so they can't, the reconstruction techniques just are not good enough yet to really isolate the individual structures down to more than just that single first unit. This is actually, on the right is actually what this same protein looks like, with the glycans there. So you can see that it's a very different picture. And this dark blue fuzz, these are the N-link glycans. Each one of these little puffballs basically represents like the excluded volume, if you will, or the volume that is sort of taken up by these glycans, which are basically sort of just like moving a lot over even the course of 1 microsecond of dynamics. So this is showing sort of the position of one glycan, each of these puffballs, over the course of a microsecond. So you can see that basically each of these glycans, they're moving around and they're basically sweeping out, they're sweeping around and moving around sort of the space. They create this sort of like dynamical fur coat that hides this molecule from the human immune system so effectively. And this was cool because what we learned, we learned not only that, this molecule, the importance of the glycans for shielding, but what we also found was that there were 2 glycans in particular, which I'm showing here. Sorry, my movie is kind of jerky. But there were 2 glycans in particular at positions 234 and, oh my gosh, how could I possibly be forgetting? Is it 165? That actually propped up the receptor binding domain of the spike protein in an open conformation, which basically indicated they were acting as part of the infection mechanism themselves. So, really important to include them is sort of the lesson that we learned. And so coming back to the flu, as I mentioned, in our original work, it didn't have the glycans, but then we later came back. These were simulations that were actually completed before the whole pandemic happened, and we're sort of we've been now coming back to those simulations to actually get them, to actually learn something from them. So this is the flu virus, and in blue are the hemagglutinin molecules, and you can see there's many of them. There's 236 on this particular version, but there's on this particular model that we have. And the red molecules here are the neuraminidase molecules. There are 30 of them. Each of these had has some end-linked glycans, which I'm showing here at these different positions. This model also contains the M2 ion channel. There's roughly 11 of those. And then the glycans here, as you can see. So I think the dimensions of this were just about 100 or 120 nanometers in diameter. Again, with all atom resolution, this is just some detailed information about the glycans that we included, but I'll just sort of skip by that. I will say, just as a sidebar, in the case of something like, when one simulates these viral glycoproteins, sometimes it's actually pretty difficult to figure out exactly how they should be glycosylated. Glycans contain a number, they're very heterogeneous, can be very heterogeneous, ordinarily are. And sometimes the profile at a particular site is very consistent, but other times there's variation in that. And so I think one of the challenges to folks like us, to molecular modelers, is really trying to make the best model that we can, or set of models that accurately reflects the biological data. And sometimes that's harder or easier to do depending on the system. With SARS-2, it was actually not as bad as or difficult as some other systems. Influenza, a little bit more variation. HIV turns out to be really difficult. But in any case, one should really try to include these. And so these simulations, so we basically built the H1N1, so the swine flu. Do you remember in 2009 there was a swine flu pandemic? That luckily, everybody caught that actually pretty rapidly, but it wasn't as severe in terms of the health outcomes, of course, as was for SARS. But so we built the 2009 strain, and we also built the strain that evolved 6 years later. So one of the interesting things about influenza is that it has this thing called a glycan clock, where you have this one particular set of strains like H1N1 that start circulating in the population, and over time, in this case we're talking about years time scale, almost all the flu variants will mutate in a way where they actually adopt a new glycan. So we call this the glycan clock, because basically every 5 to 6 years the flu will add another glycan, typically in this place where the antibodies bind. So it's due to immunogenic pressure. And then they have to redo the vaccines. In any case, so we wanted to understand some of the structural differences between these two strains. And so this is what we found. So the first thing that we noticed, so you run these huge simulations. And by the way, I guess these simulations had on the order of 160 million atoms, and we were able to run them, as I showed in the last slide, but then I didn't talk about, we were able to run them for about 500 nanoseconds in triplicate. Then you start looking at the data. And one immediate thing that we saw was that the hemagglutinin molecules appeared to be tilting. And so here you're looking in a sea of molecules. The guy who I just highlighted as blue was a hemagglutinin. What we saw was that many of the hemagglutinin appeared to tilt relative to the membrane. So here I've isolated one hemagglutinin molecule. You can see in it, which is the surface, which is showing as it gets more bent, it turns more yellow. So it goes from purple to green to yellow, I believe. And those glycans are also shown there in yellow. So you can see there's a fair amount of tilting that happens. And so we started looking at this more in terms of quantifying the tilting motion. And what we saw, so just looking at a histogram of like all the different snapshots we saw, that this ectodomain tilt angle sort of really had a wide range across both of the simulation constructs that we saw. Now, interestingly, or I think this is interesting, when you look at so most of what we see when we think about these glycoproteins, because we are informed by the experiment, when we're talking about it's atomic resolution structures, we're generally looking at either crystal structures or we're looking at single particle data. We think about these single proteins, right? You get the high resolution of like the single protein, and we always think about it, or I always thought about it as sort of like this upright system. But it turns out, as I just showed you, that actually there's a fair degree of flexibility of the domain, sort of tilting either way. And interestingly, there was one experiment of the full-length hemagglutinin in mice cells, which did in fact observe a tilt angle of 52 degrees, which I'm showing here. So there was in fact experimental data that these guys could tilt, which was pretty neat. We were happy to see that because of course, I think one always is concerned that you set up these simulations, you run them, and then maybe you're just simulating artifacts. So anyway, we were happy to see that. But so then I also want to say, and I won't go on too long about this, but one of the really, I think, also wonderful things about these mesoscale molecular simulations, I'll call them, is that you have so many copies of each of the types of molecules in the system that you can use techniques, statistics-based techniques like Markov state models, to look at the dynamics in a different way. So we were able to, if you have 236 copies of the hemagglutinin, you can extract all of those. And then we were able to determine the rate of bending. And we saw that, it takes about a microsecond or so to, for it to bend over. And then once it's bent over, returning from the tilted to the untilted state is actually rather rapid. And so, so I just say as a proof of as a general comment that, it is a terrible pain to work with these big systems. They're difficult to visualize, they're difficult to move, it makes terabytes of data that are just like a pain. But there are some advantages, including what we can discover, which I hope, in terms of the science, which I hope to convey in this talk, but also part of that is also being able to use things like MSMs. Okay, so so we characterize the dynamics, all right. But one of the things that's really interesting that we saw, in terms of this tilting motion, is that it actually it reveals what is known as the anchor epitope at the base of HA. So at the base of the hemagglutinin molecule, where it meets the membrane, is this epitope here that I'm highlighting in green, the small epitope that actually binds, is known to bind an antibody. And when if you think about the hemagglutinin and you just are simulating it in the planar, membrane, and it's sort of, just thinning in it, that epitope, that anchor epitope is actually, it's not exposed enough to allow access to or binding of that epitope. It's only through bending. So what's really neat about this is that this epitope is a target for broadly neutralizing antibodies. So there's a whole big effort in trying to develop broadly neutralizing vaccines for flu and also for things like COVID-19, of course, or for SARS-2. So this is sort of showing how these simulations actually can be used to understand the structure, dynamics, and exposure of these epitopes. Okay, so another thing that we found, again, when we were looking at the simulation, so here we're going to dive in, is related to the hemagglutinin, but not its tilting, but something that we call its breathing. And so now if you're looking at this hemagglutinin here in blue in the middle, what I hope you see is that actually the hemagglutinin is a trimer and it's actually sort of opening up. It actually breathes open during the dynamics. There it gets kind of wide, and it, there again, you can kind of see it. I'll just let you watch it. So here's another view, taking it out of the crowded environment. Now, this from the side, you can see how this trimer is breathing open, and from the top on the right, this is the top view on the right. Oops. Okay, so when we saw this, we were also thinking, this is interesting. We had simulated HA a lot as an individual particle, and we never recalled seeing, this type of breathing motion. So we started to go in there and try to characterize it more. Again, here I'm showing like the histogram of this is the hemagglutinin head center of mass distance. So to get an idea of like, how far apart they're splayed. And we can see that it's a bit more narrow, but there is some opening that we see. This gives a sense of the range of it. And then we were also able to characterize the kinetics again with the Markov state models of this breathing motion. And it's on the order of, hundreds of nanoseconds to open. And here what we're showing is looking at this what they call the receptor binding domain, also for hemagglutinin. So the distance from these top domains to the core of the molecule, and I'm showing the different traces over the course of the dynamics for chain A, chain B, and chain C. So this is what, when it's closed, this is sort of what it looks like. This is, when you pull down a molecule from any of the structural databases, this is generally what you see. This is one of the more open states here. It's corresponding to about some time point around 300 nanoseconds. And then we also sampled in one of the chains what we call a super open state, which is like really splayed far open. And so, we also started looking at this, and then in looking at the literature, something that had come out just a couple of years ago was this really nice paper by our colleague Ian Wilson and folks from Vanderbilt, and also across the street here at Scripps, was that they had seen that there was a site of vulnerability on the hemagglutinin head trimer interface, and actually that they had seen in these single particle studies and also in these immunology studies that the head did in fact open. And here you can see this open HA trimer. They have very low resolution, structural data. This would be almost what I sometimes refer to as blobology, right? But just they don't have enough images to create high resolution structures, but you can see here that it's really splayed open. And this antibody, this flu A20 antibody, was binding. And from their paper, I just want to read this one statement that they made. So, here, these are people who work, for years with these trimer molecules. And they said, therefore, the HA molecules on the cellular or viral surface generally have been considered to be stable trimers, with the trimer interface regarded as inaccessible and thus not targetable by the immune response or therapeutics, right? So let me just rephrase what they're saying. We always think about, because we can't capture their structure necessarily at high resolution, we generally consider these trimers, and it doesn't matter if it's HA trimer, it could be a SARS-2 spike, it could be an HIV envelope. All these glycodomains, many of them use these fusion proteins that have these similar trimeric architectures. We always consider them closed, and the inside bits where there are epitopes that are broadly neutralizing, that they're inaccessible. But here, just want to read this because I found this so intriguing. The ability of flu A20, that's the antibody, this broadly neutralizing antibody that they found, to confer in vivo protection strongly suggests that HA molecules are dynamic and more heterogeneous in their conformations than we have observed previously, and that the trimer interface is partially or transiently accessible. And then they talk about this thing, breathing. And so that's exactly what we saw in these simulations, right? That we're actually seeing the breathing of hemagglutinin, and this breathing reveals an opening of this that can accommodate the binding of this flu A20 antibody, which was really, I think, was sort of an exciting finding for us. Okay, and then the last thing that I want to talk about was something else now totally unexpected. Well, what about neuraminidase? I have to say, neuraminidase is one of my favorite molecules of all time. I studied it like crazy as a postdoc with Andy, and so on many years after that. And when we were looking at it, look at this guy. So this is a neuraminidase. It's sitting on a stalk. I hope what you see here happening, it's way tilted. It's like way bent over. Ordinarily we think of this thing like, I don't know, like sitting on a stalk, and then it has it's a tetramer head, but that it's just very, I don't know. I never thought about like the tilting of it, but here we see really clearly that it's not just sitting like a flower bouquet, just straight up. It's actually, it does have this, not all of them, but some of them have this really remarkable degree of head tilting. And so now we're really thinking like, oh man, is this an artifact? Like, look at this guy. He's like super far bent over. What's going on? And, so we did the same thing, right? Where we looked at all of the different head tilt angles, the histogram of that across the different strains. And, okay. And we characterized the dynamics of this head tilting. Okay. And then I was giving a talk on SARS-2 at the NIH Vaccine Research Center. And after my talk, Masaru Kanyeku basically said, I have something that I want to show you. And it turns out that they had discovered, but not yet disclosed, what they were calling a dark side NA antibody. So they had a patient who was infected with H2N2, a different strain of the flu than what we simulated, but another one that, people catch. and they took the plasma to this convalescent donor, and they saw that what they found was that there was evidence of an antibody that was binding to the bottom side of the NA head. And so here, what I'm showing in red is the neuraminidase tetramer. In orange is an antibody that binds the top part of neuraminidase. And then here is showing the evidence, the structural evidence of this other, what they call neuraminidase dark side, NDS.1 antibody. And so, so you can see that here. And so this is the top view where you can see the one they knew about, and the bottom view where you have these sort of clouds. So this was really neat, because they were excited to see that these simulations actually showed how this antibody or this epitope could become accessible to these antibodies. This was the work of Julia Lederhofer and Masaru Kanyekiu, who I was mentioning at the VRC. And so that was sort of exciting that, and of course, we're always happy when when these things that we see, and this is so often the case, right, that sometimes you see things in simulations and you think, I don't know, I think, did we really mess this one up? Maybe we forgot to include that disulfide bridge, or, whatever it is. But here, actually, to see that these motions, could be further corroborated by this experimental data was really exciting. So this was in this is disclosed in a paper that's now in the bioarchive and currently under review. This is just a higher resolution image, but this is sort of a guess at, because they don't have a high resolution structure of the dark side antibody. So this is using a different model of the Fab, but you can see how the Fab would fit into the density envelope that they see there. Okay, and so I will close so we have some time for questions. It's about 47 minutes by my clock. So in terms of acknowledging, of course, I have just a terrific group here in La Jolla. There's just so many wonderful people in it. I think we have such a diverse group from so many viewpoints, and I'm so grateful for my group. They're just so fantastic. And also I'm hiring, so if folks are interested to do this type of work, we have a lot of work right now trying to do small molecule computer-aided drug design for a number of different emerging viral targets through one of these large awards from the National Institutes of Health here. So if anyone's interested, please do reach out. And we also, of course, we do have a lot of work also now in what we call structure-based vaccine design, more like what I just told you about with the flu. But I also want to thank, of course, Lorenzo Casalino has been someone who really has led the simulations of not only the influenza virus with the glycans, but also all the stuff with much of the work with SARS that you heard about, or that you may have heard about, that Phil mentioned at the beginning. Christian Seitz, another graduate student in the group who is getting ready to graduate and who led the Markov state modeling for this project. Sarah Kahanik and Jacob Durant for their early work in being very brave to build that 160 million atom system. It's really hard work, and it takes years to simulate and then years to look at the data. But I think now we're to the point where the timescales will get faster for that type of turnaround, and hopefully we show that it's worth it to do it. Of course, and then our experimental collaborators for the flu in particular, Julia, Masaru, and Ian, funding and so forth. And I just want to also give a shout out to Frank Noe, Dan Zuckerman, and Huancheng Zhou, who have been helping us to, understand better the limitations of and the applications of Markov state models, as well as these crowded or confined systems. All right, so that I'm happy to take questions.